Hi, I would like to give you a brief introduction into what we do in the laboratory. We are absolutely fascinated with the brain and the complexity of the brain, with its 80 billion neurons and its intricate connectivity. We are trying to study the brain at several different levels, ranging from the study of single neurons and its input-output properties to the study of circuits where several neurons are aggregated into recurrent motifs. Secondly, we are interested in mechanisms of CNS disorders, and we are absolutely convinced that CNS disorders can only be understood at the circuit level. So we are applying all these techniques to the study of epilepsy, one of the most common CNS disorders. We've assembled a great team to address these questions at all these levels, and so I would like to take you on a tour through the lab.
My area of interest is in investigating how neurons transform synaptic input into an appropriate output. And more specifically into how their dendrites, their site of input, actually act as computational units. To tackle these questions, we use some sophisticated electrophysiological and imaging techniques that employ 2-photon pulsed lasers. We firstly patch onto cells, fill the cells with a fluorescent dye, and then we can actually stimulate individual synapses using 2-photon uncaging of glutamate.
In our lab, we're interested in how the hippocampus computes information. Therefore, we investigate networks and network patterns in vivo. To do so, we perform 2-photon calcium imaging in neural ensembles. This allows us to gain deeper insights how different states of brain activity are correlated to behavior.
Having all these different techniques in mind, which we perform here in the lab, one could think of an implementation in medical research. Epilepsy is a severe disorder which finally causes seizures in patients. It is our interest to investigate these seizures on a network level. And for this purpose, we are using electrophysiological and pharmacological approaches. We can investigate how different anti-epileptic drugs act on a network level in the brain. And we want to know why different anti-epileptic drugs work in some patients and why some patients do show a pharmacoresistance.